Planar multi-electrode arrays are used to record from the vomeronasal organ. The VNO is removed from the mouse such that the neuroepithelium remains intact. First, the VNO capsule is removed from the mouse, then the neuroepithelium is isolated from the bony capsule, blood vessel, and basal lamina. The tissue is placed in the MEA with the dendritic knob side up. Once this setup is complete, we use a robotic arm to administer liquid stimuli to the tissue.
Hi, I'm Hannah Arnson from the laboratory of Tim Holey in the Department of Anatomy and Neurobiology at Washington University School of Medicine.
I'm Xiaoyan, also from the Holey Lab.
Today we'll show you a procedure for multi-electrode array recording of the vomeronasal organ.
We use this procedure in our lab to study the sensory properties of receptor neurons in the accessory olfactory system.
So let's get started.
Electrical recordings are obtained with a planar multi-electrode array, or MEA, with 10 micrometer flat titanium nitride electrodes insulated with silicon nitride. 60 electrodes are arranged in two fields, consisting of a 5x6 grid with center-to-center spacing of 30 micrometers. Using an MEA 1060 amplifier, the electrical signals are amplified 1000-fold. Signals are acquired at 10 kHz using a data acquisition card and custom data acquisition software. The tissue is held down on the MEA by a nylon mesh, which is attached to a custom insert which fits snugly inside the culture tube ring of the MEA. Now that we've seen the MEAs, let's see how to do the VNO dissection. Prior to performing the dissection, fill the MEA well with bovine serum albumin.